Okay, in our last lecture of the DNA chapter, we're going to look at the second part of gene expression, which is called translation. So let's get going. The due now for today's class was to describe what mRNA does and why it's. So essentially, what the idea is that mRNA copies one gene from your DNA code, and then it goes to the ribosome and tells it which protein to make. This allows the DNA to remain protected in the nucleus, and it also the DNA molecule is so big that it would be really, really complicated for the whole thing to leave the nucleus just to translate one little section of the molecule of the gene into a protein. All right, so let's review what's going on. The four main topics for learning DNA are its basic structure, the nucleotide, how and why it packages itself inside the nucleus, how it replicates, and how DNA gives directions to the cell through transcription and translation. So we looked at transcription in the previous lecture. Today is about translation. All right, just to recap, RNA polymerase is the protein enzyme that reads the DNA template strand and creates mRNA using complementary bases. This creates a new strand that is identical to the DNA coding strand because the RNA strand is a complement of the template strand. The only difference is instead of thymine as a base, it uses uracil. All right, so let's start translation. At its core, translation is a process that takes place at the ribosome, and it's going to translate mRNA 3 nucleotides at a time to bond amino acids into proteins. Another way to describe 3 nucleotides is as a codon. Codon is just 3 nucleotides. Here we have 4. 1 codon, 2, 3, 4. And each codon tells a ribosome to make, or not so much to make, that's kind of a bad way to say it. We're going to say each codon tells a ribosome to assemble the next amino acid. So I'm going to change this actually. I'm going to write assemble. Assemble the next amino acid. One important thing to note is that a ribosome is not going to begin or stop making proteins or assembling proteins, assembling amino acids, until it receives a start or stop codon. So one way to decide, or one way to determine how a codon is going to translate into an amino acid is this nifty little sheet right here. The inner ring represents the first letter of a codon. This middle ring represents the second letter of a codon, and then the last outer ring represents the third letter of a codon. If you follow it from start to finish, you'll end up within an amino acid. So for example, UGC will code for cysteine, and that's exactly what that is. CAA will code for glutamine, CUG will code for leucine, and interestingly enough, UUG will also code for leucine. Because remember, there's only 20 amino acids, but there are many, many more combinations. So there's going to be some redundancies. Okay, let's look at the key players of translation. The first one is the ribosome. This is where it all happens. This is the organelle where proteins are made. They're going to assemble amino acids into a long chain, and that long chain is called a protein. Amino acids are the building blocks of proteins, and there are 20 of them. They're bonded together with a special kind of bond called a peptide, and many peptides make a polypeptide. Polypeptide is really just another word for a protein in some sense. tRNA is another major player here. This is another RNA molecule that's going to bring amino acids to the ribosomes. They have at the bottom, if you look at the anatomy here, the sphere is supposed to represent an amino acid. The rest of the body kind of is allowing a bond between some RNA down here and the amino acid up here. And down at the bottom is something we call an anticodon. An anticodon is going to have 3 bases, and they're going to complement 3 other bases. So, for example, on an mRNA molecule, this will match up with a codon that spells out G, C, and not thymine, but uracil, so GCU. With this codon, you can then use that chart from the previous page and determine what amino acid it's going to code for, and it actually is going to code for alanine. So alanine, this amino acid is actually alanine. So what happens? As the mRNA is fed into the ribosome, it's going to read this codon. So for example, the codon that's being read right now is UUU. When UUU is read, a molecule of transfer RNA, tRNA, that matches up with UUU, in this case AAA, is going to fly into the ribosome and detach its amino acid. So this amino acid would be UUU, which is phenylalanine. So this one is phenylalanine. The next amino acid that's in line to be made into this protein is going to correspond with the codon AGC. And AGC is serine. So this amino acid is serine. This codon will match up with this anticodon, and that's how the tRNA molecule knows which one is coming next. So there's going to be another amino acid somewhere over here that'll fly in and match up with whatever codon is written right here. It looks like this tRNA molecule, which has the anticodon CCC, has already dropped off its amino acid, probably this one. Now this one, remember, doesn't match up with the anticodons on the tRNA molecule. It matches up with the codon on the mRNA molecule, so GGG. GGG is glycine, so this molecule is most likely glycine. Here's this video again. It was shown in the last lecture, and it does a great job of representing what happens, how this works, and kind of in a nice computer simulated version. I love my analogies, so here's kind of a neat analogy if you wanted to compare how a protein is made to how a meal might be made in a kitchen. So your ribosome is what makes a protein. It kind of assembles everything just like a chef would assemble your meal. Amino acids are what make up a protein, just as the ingredients are what make up a meal. Your tRNA goes out and buys the amino acids, or gets the amino acids, just like a shopper would go out and buy the ingredients. And your mRNA is going to be the recipe, the instructions on how to assemble the amino acids, or your recipe on how to assemble the ingredients. This brain pop, the brain pop on RNA, is probably the best brain pop video I've ever seen. It's incredibly detailed. I would highly recommend watching it to give you a recap of everything that we just talked about. And that should end our discussion on the DNA chapter. Thanks for watching.